Without a doubt, one of my very favorite parts of teaching biology is genetics. And you can't start genetics without talking about the father of genetics, Gregor Mendel. Mendel was an Austrian monk, and he studied genetics back in the 1800s. He's kind of like the ultimate rock star or movie star of genetics, I guess you could say. But he published some papers in which he discussed how inherited traits were passed on. He worked mainly with garden peas. And you can see here there are different traits that Mendel looked at. Round versus wrinkled seed, for example. One of the most famous, of course, is tall versus short plant. Tall being dominant over short. Here are some examples of human traits. For example, free earlobes are dominant over attached. Widow's peak is dominant to no widow's peak. Dimples are dominant to no dimples. Chin cleft is dominant over no chin cleft. Hitchhiker thumb is recessive to a straight thumb. Bent pinky is dominant over a recessive straight pinky. The hair on your knuckles, mid-digital hair, is dominant to no digit hair. Second toe being longer than the big toe is dominant. Tongue rolling. And Vulcan versus Earthling. So how traits are passed on is kind of random, just like you'd roll a die to get a random number, and then roll another one for the other parent. When you roll 2, you simulate what would happen between two parents, and when you roll them multiple times, you're simulating what you would get in multiple offspring. The first type of inheritance we want to look at is called complete dominance. That's where one gene is totally dominant over the other. In this case, the wire hair is dominant over the smooth coat. When you cross two parents who are both heterozygous, in other words, they both have wire hair. However, they carry a little w. That's the gene for smooth hair. So in this case, you see that the one offspring is indeed a smooth head coat. In another example of complete dominance, we have, as mentioned before, free earlobes versus attached earlobes. In this example, two parents are crossed, one of which has free earlobes, and the other parent has attached earlobes. However, since the dominant parent does carry a attached gene, they have a 50% chance of having a child with attached earlobes, as well as a 50% chance of having a child with attached earlobes. Up next is codominance. That's where both genes are dominant. In other words, they both show up if they're present together. You see this in the example with the cats. Gray and black are both dominant. So when we cross two tabby cats together, as we call them, a cat with both black and gray, you're going to get all three in the offspring. You get one gray cat, one black cat, and then 50% of the offspring, or 2 out of 4, are going to be the tabby pattern. Black and gray together. In incomplete dominance, neither gene is totally dominant over the other one, so they blend together if they're both present. So here we have blue bird and yellow bird. So we're crossing one parent that's blue and one that's yellow. So as it turns out here, all of the offspring will be heterozygous. That's big B, little b, one of each allele. And they will be green birds. 100% green offspring. Once again, the different traits are blended together. Neither one is really dominant over the other. In sex-linked traits, sex plays a role, obviously, as indicated by the name. In this example, we're crossing a red-eyed female with a white-eyed male. 2 of the offspring will be female, and 2 of the offspring will be male. However, let's take a look at whether or not they have red or white eyes. This offspring is female, red-eyed. Here's a female white-eyed. little r. Here's a male with red and one male with white. All this variation we're seeing in these genetic traits leads back to the fact that DNA has to be copied, and sometimes it gets mutated, and that's what leads to the variety. But let's take a look at DNA replication a little bit. DNA would take a long time to replicate if we just went from start to end, so it starts at different spots called replication forks. So it may start at hundreds or thousands of different places along different points in the molecule. This takes considerably less time to do. It's kind of like when you play golf. If you've ever been part of a shotgun start in golf, that's where some people start on hole 1, some start on hole 2, and so on. Here's an example as these men get ready for a shotgun start. Some of them will be going to each of the 18 holes so that things go faster. If they all started on the same hole, it would take forever to get everybody started. By starting at different holes on the course, everyone gets to start at once and move their way through. It works the same way with DNA. When DNA is copied at multiple points at the same time, it goes much quicker. Now we turn our attention to mutations. Generally, mutations are a bad thing, period, because they alter proteins, and when a protein has the wrong shape, it can't do its job and it causes problems in the body. Things that can cause mutations are UV light from this tanning bed. You also get UV light from the sun. Either way, that UV light can cause mutations. This is asbestos. It used to be used for insulation. It is also an example of a substance that can cause mutations. And of course, smoking. Cigarette smoke contains many different types of substances that can cause mutations. So here's the deal. When you get a mutation of a protein, it alters its shape. We talked way back in the beginning of the video about the importance of the shapes of proteins and the job that they do. So in this case, protein A is the correct protein. Protein B has a dysfunction, which means it cannot do the job it is meant to do. It's like this car. Everything is perfect on it except for not having any wheels. So even if the motor and everything else is in perfect working condition, without the tires and wheels, it's not going anywhere. By the same token, a protein with just one small mistake can't necessarily do its job. So here's what mutations cause. Like we said, proteins that are misshaped and therefore faulty. They can't do their assigned job correctly. This leads to things like diseases and disorders, anything from cystic fibrosis to Huntington's to muscular dystrophy, you name it. And the big cancer problem is due to mutations. Mutations in certain genes cause cells to grow uncontrollably, leading to cancer.